So welcome back. We are in the middle of chapter 13. This is the second lecture for chapter 13. We are talking about meiosis. And we've already talked about kind of a basic what sexual reproduction is. We talked about some different sexual life stages. And now we're going to get a little bit more into what is actually occurring in meiosis. So before meiosis happens, you have chromosome replication, the same as what we see in mitosis. So your cell is going through, growing, copying its DNA, and then it's going to get into actually going through meiosis. Meiosis is going to have two sets of cell division. So you copy your DNA once, then you divide your cells in meiosis 1, and then you divide your cells again in meiosis 2. So because you've divided once, that gave you 2 cells. Then in meiosis 2, you went from 2 cells to 4 cells. So at the end of meiosis, you have 4 daughter cells. So daughter cells means the 4 cells you end up with at the end. So mitosis gives you 2 daughter cells that are identical. Meiosis gives you 4 daughter cells. And because you only have 1 round of DNA replication, in meiosis 1, you end up with 1 copy of every chromosome. In meiosis 2, that is when you become a haploid cell where you have half as many chromosomes as a parent cell. So the chromosomes are going to duplicate before meiosis during interphase. 1 copy of each chromosome becomes 2 copies of each chromosome. And then these sister chromatids are closely held along their lengths. Cohesion is holding them together. And so this is sister chromatid cohesion when they're held together. And by meiosis 1, you have duplicated chromosomes, 1 of each. So your dad's chromosomes are going to 1 and your mom's chromosomes are going to the other cell, but they're still both duplicated. You have 2 copies of that gene. And then meiosis 2, your sister chromatids are going to separate when you have 4 haploid cells. So instead of having a copy of every chromosome, you have 1 chromosome or the other. So you have your dad's chromosomes or your mom's chromosomes. So you're a haploid cell here, you're a diploid cell here. So division in meiosis 1. So meiosis 1 is the first one. This is going to happen in 4 phases. 4 phases: prophase, metaphase, anaphase, and telophase. And because these are in meiosis 1, we're putting a 1 after the end. So it's prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. In prophase 1, you have copied your DNA already, so you have sister chromatids held together. You are forming these spindle microtubules. Your nuclear membrane is dissolving. In metaphase 1, those sister chromatids are still attached to each other, and you can see you have both copies of chromosome 1 from your mom and your dad. They need to be lined up together. And for each chromosome. So you not only have your sister chromatids together, but you also have the chromosomes from both parents lined up together. At metaphase 1, you have this metaphase plate. At anaphase 1, you are ripping those apart, but you can still see sister chromatids are still held together. So the dad's copy goes one way, the mom's copy goes the other way. And then telophase 1 and cytokinesis is when you are breaking into 2 cells, reforming your nuclear membrane. And so now each cell at the end of meiosis 1, each cell has sister chromatids, but they should have sister chromatids for every single chromosome. So these 2 cells are not identical, right? And then prophase 1, each chromosome pairs with its homologue. So you have homologous chromosomes put together. Crossing over can occur. We haven't talked about crossing over yet. But crossing over is why you see here like a blue chromosome, your dad's chromosome with a little red piece on it. Here's a red chromosome with a little blue piece. That is because ends of these DNA can actually swap places. So you end up with chromosomes that aren't totally from your dad or totally from your mom, but a mix of genes from your dad and mom, both on that 1 chromosome. And then we're going to form chiasmata between 2 different chromosomes to have crossovers. Chiasmata is probably not a term I'm going to ask you to describe to me. Your textbook goes into that a little bit more. We may touch on it a little bit later in this lecture if we get around to that. In metaphase 1, you have not only sister chromatids, but also homologous chromosomes lined up together, joined together, lined up at that metaphase plate. One chromosome faces each pole. So one homologous chromosome faces one way and this homologous chromosome facing the other way. It doesn't have to be all moms on one side and all dads on the other side. It's kind of random which side has the mom's chromosome, which side has the dad's chromosome. And then by this point, there's already been crossing over, so it's not entirely mom's chromosome and dad's chromosome. Your microtubules are going to attach to the kinetochores of the chromosomes. So one microtubule from this side attaches to the chromosome here. Microtubule from this side attaches to the chromosome here. That anaphase 1, those homologous chromosomes separate. So the sister chromatids still attached, but homologous chromosomes have now pulled apart to opposite sides. And that's the spindle apparatus that is actually like pulling them apart, those microtubules making them move, as those microtubules shorten and pull the chromosomes along with it. Then at telophase 1, it starts with each half of the cell having a haploid set of chromosomes, so 2 copies of that DNA, but it is haploid at that point because it only has from your mom or from your DNA. You don't have 2 distinct copies of each gene. You have 2 replicated copies of each gene, if that makes sense. And if it doesn't, let's talk about that some more because I'm not sure that I'm explaining that the most clear way. But you can watch some of these supporting videos, read your textbook, and talk to me anytime something isn't clear. At the same time of telophase 1, you have cytokinesis. So you can tell we have this cleavage furrow. This is an animal cell dividing these 2 cells individually. Haploid, but with replicated chromosomes. So still sister chromatids there. So in animal cells, you're going to have this cleavage furrow. In plant cells, you would have a cell plate forming. And then it is going to jump right into meiosis 2. It is not going to copy DNA. It's not going to do anything else. It already has replicated chromosomes, so it's jumping right into meiosis 2. In meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. And this is going to happen very similarly to what we see happening in mitosis, except we're doing it with a haploid cell instead of a diploid cell. So in prophase 2, we're forming our spindle apparatus. You can see here, this is your spindle apparatus. And your chromatids, which are still sister chromatids, are going to start moving towards the middle of the plate. In metaphase 2, you are lined up at that metaphase plate. And so this is, since we've already divided into 2 cells, we're going to keep following these 2 cells. In metaphase 2, your DNA, your sister chromatids are lined up at the metaphase plate, connected to the microtubules at the kinetochores. In anaphase 2, the sister chromatids are separated. So those cohesins holding those sister chromatids together are separated, and the individual genes move towards opposite poles. And then in telophase 2, you are going to have your chromosomes at opposite sides of the cell. Your nuclear membrane starts reforming, and chromosomes start decondensing, turning back into loose versions that can be used to do whatever work that cell needs to do. And you've got cytokinesis separating the cytoplasm, either by a cleavage furrow or a cell plate again. And so at the end of meiosis, you'll have these 4 distinct cells. And each cell is going to have a unique combination of genes from the mother or from the father. You're not identical. Almost always, you have 2 cells that are never going to be identical. So each daughter cell, each oocyte, or each sperm is going to be genetically distinct from the parent cell. So that's why, even brothers and sisters can have very different genes, because each egg cell and each sperm cell is going to have a very different, unique combination of genes in them. So let's watch this little Bioflix clip.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red. and the other set is from their father, shown in blue.
So I just want to throw in here, this is probably obvious to you, but the chromosomes from mom and dad are not actually going to be different colors. They're going to look all the same. We're just coloring them red and blue in the picture to make it easy to follow what's happening in this process, okay?
Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of two identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of two sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into two cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay, so my hope is that you would be able to explain each step of what's going on in that video just independently. Take a blank sheet of paper and be able to explain all the steps of what happened there. So I want you to, at this point, pause this video, see if you can explain all of what's happening through each step of mitosis, all of meiosis 1, all of meiosis 2, and then go back and watch that video again and see if you need to update, see how much you really understood and remembered and where you need to kind of work on studying what's happening again. Okay. Give me a second here. So I am going to pause this video at this point, and we'll come back and talk about some of the ways that we get variation happening as a result of meiosis, how we have genetic variation through our species. So come back for part 3 and we'll discuss that in its own part. I'll see you in a little bit. Bye.